Now, today we are going to be getting into the process of meiosis. So, we are truly going to begin to break down how meiosis goes through division. So, just a quick recap for you guys. If you recall, there are two types of cells in our bodies. We have sex cells, which are known as your gametes, and you have somatic cells, known as your body cells. Now, there are two types of chromosomes that are also going to be found in our cells. You have autosomes, which is what controls inheritance of traits, and you also have your sex chromosomes, which control your sex, which is if you're genetically male or genetically female, and any sex related traits. Now, meiosis is the process by which gametes are formed. So, this only happens in sex cells. It reduces the chromosome number by half, which is N for haploid, because it produces haploid cells. Now, when gametes combine to form the zygote, the original chromosome number is going to be restored. So, here is the process of meiosis. So, meiosis is divided into two phases. We have meiosis 1. Meiosis 1 will separate the homologous chromosomes into mom's set as a separate set and dad's sets. Then it goes into meiosis 2. So this is what separates the sister chromatids, which mom's set gets split up and dad's sets will get split up. So during meiosis 1, we have interphase. So the G1 part of interphase is cells will grow and perform many of their required cellular functions. And we already know that during S phase, DNA is copied or it's replicated. Now, in the G2 part of interphase, that is when proteins are made and they grow. Now, we have prophase. So, prophase 1 is when the nuclear membrane is going to break down. So, the centrosomes are going to move to opposite sides of the cell. And that is when the spindle fibers will start to assemble, and you'll see those later on. So the duplicated chromosomes are going to condense, and the homologous chromosomes are going to pair up. So here, these are our spindle fibers. The biggest thing when I'm talking about this is watching the pictures to truly understand what's going on. Now, we have metaphase 1. This is when homologous chromosomes are going to pair up randomly when they're lined up in the middle of the cell. So chromosomes, some from the father and some from the mother, are going to line up along each side of the cell's equator. So next you'll have anaphase 1. So this has the paired homologous chromosomes. They separate from each other and they are moved toward opposite sides of the cell, while the sister chromatids are going to remain together. And finally, we have telophase 1. So this is when the nuclear membrane is going to form again in some species. The spindle fibers are going to disassemble and the cell undergoes cytokinesis. So the end result is that two cells that each have unique combination of 23 duplicated chromosomes coming from both parents. So this is what it's going to look like in the beginning, and it's going to go through that process of separating until you have two cells. So let's talk about genetic diversity and variation. So there are two mechanisms that occur during meiosis 1 which introduce genetic variation. Genetic variation is very, very important. That's what makes you different from your siblings, even if you have the same mother and father. So, one thing is crossing over. This happens when homologous pairs will line up opposite of each other at the equator of the cell. So, parts of the chromatids can become twisted around each other. So, this can result in a new combination of alleles. So, chromosomes can transfer their genetic information and exchange it between each other. So let's say this is how they looked originally. But when crossing over occurs, this has caused a little bit of the purple to be on this side and a little bit of the blue to be on this side. So it is a new combination of alleles due to that crossing over that has occurred. Something else that can happen is independent assortment. So, independent assortment is when homologous pairs line up opposite of each other at the equator of the cell. So, it's random which side of the equator the paternal and maternal chromosomes lie. The paternal chromosomes come from dad, while the maternal come from mom. So, these pairs are separated, so one of each homologous pair is going to end up in the daughter cell. Now, this is going to create a large number of possible combinations of the chromosomes in the daughter cells that are produced. Now, let's get into some probability and math for a second. So, we have two different possibilities here. We have possibility 1, possibility 2. So they are two equally probable arrangements at metaphase 1 that can give rise to different chromosome combinations during metaphase 2 and your final gametes. So you can see, even though we're starting with the same things, we can have so many different combinations of cells, which is what gives us our genetic diversity. Now, we have meiosis 2, which is going to start with prophase 2. So, the nuclear membrane is going to break down, and the centrosomes are going to move to opposite sides of the cell. And that is when spindle fibers are going to assemble. So, notice that with meiosis 1 and meiosis 2, our steps are going to sound pretty similar. So, next you have metaphase 2. So the spindle fibers are going to align chromosomes at the cell equator. So each chromosome still has two sister chromatids. So next, you're going to go into anaphase 2. So the sister chromatids are once again pulled apart from each other, and they're going to move to opposite sides of the cell. And this is going to lead to our telophase 2, which is when the nuclear membranes are going to form around the chromosomes. Your spindle fibers are going to break apart, and the cell is once again going to undergo cytokinesis. So the end result of meiosis 2 is you are going to have 4 haploid cells with a combination of chromosomes from both the mother and the father. So you start with a single diploid cell, and this is the process as a whole, which is going to end with our 4 haploid cells. Something that has always helped me out, you don't have to do this, is just going through each individual step and drawing it out for yourself. So you can watch the movement of the chromosomes within the cell so you can understand how they're moving and how they're breaking apart. Now, we have meiosis versus mitosis. So, mitosis It creates all of the cells in our body except for our sex cells, while meiosis uses sex cells only, so the egg and the sperm. Now, the definition of mitosis is that it is the process of cell division that forms two new daughter cells, each with the same number of chromosomes. While meiosis, while it's also a process of cell division, it has four new cells that are created each with half the original number of chromosomes. So mitosis will only go through one round of cell division, while meiosis will go through two rounds. So since meiosis has nothing to do with sex cells, it is going to go through asexual reproduction, while meiosis will go through sexual reproduction, since it will require the assistance of the egg from people who are biologically female and the sperm from people who are biologically male. So your end result for mitosis is going to result in your two diploid cells, which are your 2N, while meiosis will result in your haploid cells, which is N. So mitosis will have no genetic diversity, while meiosis does, because it has the introduction of paternal and maternal cells. Now, the purpose of mitosis is to repair the damaged body cells through growth, while the purpose of meiosis is to create sex cells that are needed for reproduction. All right. That is our overview of mitosis and all the different stages of meiosis. Make sure that you're filling out your sheet to keep up with the steps. And if you need any help, just let me